Hi, you guys. I am here virtually because I'm not live in class today, and I'm going to give you guys a quick little chalk talk introduction to the food dye lab. In my experience, this is one that works a little bit better if I break it down for you guys before we get started, because there are quite a few moving parts, and it's pretty easy to get confused on this one, but I got you covered. I'm going to walk you through it, and I will be around if you guys have questions. I'm expecting them, so please don't hesitate to ask. I got you covered. So with that, our food dye lab, you'll see that in your lab archives, is going to be based on the principle of UV vis spectroscopy. What that means is we are going to use UV, so ultraviolet and visible light, and a technique called spectroscopy, which is a whole range of experimental techniques that give you information about what's in your sample. And so we're going to use UV and visible light to tell us about what is in some sample that we have. So before I start talking about the actual experiment, I want to tell you guys a little bit about what this actually is. So first things first, you guys are going to have an instrument in lab called a spectrometer. And what that's going to be is a box on your benchtop. And there's going to be like a space in that box for you to put a sample in. So you will have a box and you will have some sample, and you're just going to stick your sample into the box. Your box is going to be connected to your computer. And your computer is going to make you a really nice graph, and that graph is going to have all of the information that you need on it for this lab. Okay, but how the heck does it make that graph? Great question. I'm glad you asked. So inside of this box here, what's going to be happening is first, we have some light source, and it's going to spit out a bunch of different wavelengths of light. So I've drawn a sun. It's really going to be a light bulb, but I can't draw those. So your light source is going to spit out a bunch of different wavelengths of light. Okay, so pretend these are all different wavelengths of light, or different colors. When I say wavelength, you can understand that as color, right? A different wavelength is going to be a different color. If you look at your electromagnetic spectrum, that's where you get the rainbow from. So I'm going to use this Greek letter, kind of a funny squiggly looking thing. That's called a lambda. And we're going to use a lambda to represent wavelength. So maybe I have wavelength 1, wavelength 2, wavelength 3, and so on and so forth. So there's going to be a whole bunch of wavelengths coming out of this light. And then we're going to have something that I call a wavelength selector. And I'm going to draw it as a triangle. So a wavelength selector. And a wavelength selector is going to do exactly what it sounds like. It's going to choose one of these wavelengths that you put into it, and it's going to spit out exactly one of them. So maybe we only spit out this second wavelength here, right? And it's going to block the rest of them. And if that seems kind of hard to understand, think about like when you shine a flashlight onto a prism, right? If you ever played with a prism when you were a kid and it spread out that light and made a rainbow, a prism is actually a type of wavelength selector. There's other ones, but think of it like a prism for now. It basically makes a rainbow and it only sends one color from that rainbow in the direction that you want it to go. So now that we've selected the wavelength that we want, this light is going to interact with our sample. So I'm going to put our sample here. It's going to be in a little sample cell, so sample. And let's say the light that we have going in is 100% of our light, right? So we have 100% of whatever kind of wavelength 2 is. Maybe coming out of our sample, we're going to say, now we still have wavelength 2, but now we only have 60% of what we put in, right? We started with 100% and now we're at 60%, right? So we have 40% of our original light and that went somewhere. That 40% of our light got absorbed by the sample. So 40% of wavelength 2 got absorbed into your sample. And so then I'm going to draw a really uncomfortable looking eyeball because I am not an artist. Okay, and so then in the instrument, we have something called a detector. And what the detector is going to do is just measure how much light we put in versus how much light we got out. And it's gonna say, okay, I noticed that that's less light coming out. And it's gonna tell you like 50% of your light came out, 60%, 70%, and so on and so forth. So remember, all of this right here, this big diagram, starting with the sun and ending with the detector, all of this is happening inside of your instrument. So you're not physically doing any of this. What you are going to be doing in lab is picking up a sample cell, putting it into the machine in a little spot in there, and then you're going to hit go on your computer, and your machine is going to do all of this for you. But if you're curious, that's what's going on inside of that instrument. So what the heck are we going to do with this information? Let me erase this, and then I will go ahead and break down this week's lab. So with this week's lab, you are going to have some kind of sports drink. So you guys will need to bring in some kind of drink into lab. And this is probably the one and only time I will ever tell you to bring a drink into lab. So here's my pretty rough attempt at drawing a beverage. So you are going to bring some beverage in. And let's say this beverage that you bring in, maybe you bring in a Gatorade and it's green. Okay, so we know that if we have something that's green, it's composed of blue and also yellow. Our goal for this lab is to figure out the concentration of blue and yellow, or whatever two colors are in your drink, right? If you have a purple drink, you're going to have blue and red, something like that. Our goal for this lab is to figure out the concentration of these two dyes in this drink. So you're going to have a drink with two food dyes in it, and you want to figure out what the concentrations are. So we want to know the concentration or molarity of blue and the concentration or molarity of yellow in our green drink. So we don't know these two right now. In order to do that, we're going to use UV spectroscopy. So you are going to, on the back of your drink, where you have your nutrition facts, it'll tell you, like you have red dye number 40 or blue dye number 1, something like that. We are going to have what we call standard solutions in lab. And with these standard solutions, we'll have, okay, this green is not showing up very well. So pretend that this was actually purple. We're going to have a solution of blue, and we're going to have another solution of red. And these are just going to be things that we already have in lab. So you're going to have some drink. I will redraw this for you guys. And we're going to say it's got some red in it, and it's also got some blue in it. And then in class, we're going to have things that are just blue and that are just red, right? So maybe you have red dye number 40 and blue dye number 1. And so we are going to make what we call a calibration curve for each of these dyes. And that calibration curve is going to allow us to figure out what's actually in the drink that we have. And so when you put your drink or one of these dyes into your instrument, your spectrometer, you are going to get out a graph that looks something like this. Okay. And what this is telling you, what's on these axes, on the x-axis here, we're going to have wavelength. That's lambda. And you're going to go from about 300 nanometers to about maybe 800 nanometers. And 300 to 800 nanometers is roughly the range on the electromagnetic spectrum of visible light. So red, orange, yellow, green, blue, indigo, violet. That's going to be 300, 800. On this y-axis here, what you're going to have is absorbance. And absorbance is kind of arbitrary. It's going to go from 0 all the way up until 1. So if your absorbance is 0, that means that your sample absorbed 0 light. And if your absorbance is 1, it absorbed 100% of the light you put in. So if your absorbance is like maybe 0.5, that's telling you it absorbed about half of the light you put in. And so when you see something like this is going to be like for your beverage. When you put in your beverage is composed of 2 different colors. So you'll get out a graph that looks like this, where you have 2 peaks, and those 2 peaks correspond to 2 different wavelengths, right? Wavelength 1 and wavelength 2. So you have 2 different wavelengths where your drink is going to absorb really, really well. And those are going to be those 2 colors in your drink. And so we're going to use that information in order to figure out what the heck is happening inside of our drink. So let's say we know we have blue dye number 1 and red dye number 40. And we have, this is just a regular drink spectrum right here. Well, maybe if we cut the concentration of our drink in half, so we dilute it, we add some water. And so now, and you can just think about this visually, right? If you pour a bunch of water into something that's a really dark food dye color, that food dye color is going to get lighter because more light is able to go through it. And it kind of makes sense if you think about it too, right? Because if you have a dye molecule and it's absorbing light, if there's more dye molecules, something can absorb more light. So there's a higher concentration, it absorbs more. And if there's less dye molecules or there's a lower concentration, then it absorbs less light. And so, sorry, I had to check something on the screen really quickly. So let's say I cut the concentration of my dye in half. Well, I know it didn't absorb a lot of light down here, right? But maybe where it absorbed a lot of light, if I cut the concentration in half, I'm going to absorb way less light now, right? Oh, that's hard to see on the screen. So now we only absorb a little and we come back to the same place. And then over here, we cut our concentration in half again. So our absorbance doesn't go up as high, right? So here, where we had just a little bit of drink, maybe our absorbance was at about 0.6. But here, where we had a lot of drink, our absorbance was all the way up at 0.1. And that's because we diluted it. So what we're going to do is make calibration curves that aren't from the beverage, that are from the known blue and red dyes, and use that to figure out where on this axis certain concentrations are. So let me erase this, and then I'm going to walk you through how to make calibration curves. Okay, so I'm going to walk you guys through how to make calibration curves for one type of dye. I'm going to do this for the blue dye. You'll do the same thing for both. So let's say we have we make a solution of 1 molar blue dye. So we have a solution of blue dye and it's 1 molar. So maybe it's like a slightly faint blue. And then we can make another solution of 2 molar blue dye. And that one's going to be a little bit of a darker blue. Right? So I'm going to put some more lines to indicate that it's more shaded in here. And then we make a third solution of 3 molar blue. To make a calibration curve, you need at least 3 different solutions. If you have time, I recommend that you do closer to 5, but 3 will do. Okay. So we can see as we get more concentrated, we've got more blue color. So what we're going to do is we're going to put all 3 of these solutions into that spectrometer, and we're going to get a graph for each of those. So remember, this is going to be wavelength, and this is going to be absorbance on all of our graphs. So let's see, absorbance, wavelength, wavelength, absorbance. So maybe we see our blue peak about here. And so when we have 1 molar blue dye, we get a graph that looks like this. And if you only have 1 color, you should only see 1 peak, right? And we're going to call this lambda subscript B for blue, right? And then now we can go and we can take our spectrum for the 2 molar blue dye. And we're gonna see, again, something similar. It's gonna be at the same place, lambda B for blue dye, but now we see our peak is a little bit taller. And it makes sense, right? Because we have more blue molecules to absorb the solution in here. And so we have more blue molecules to absorb light, so we get more light absorbed. And maybe we do this again a third time for our 3 molar blue. And now we see a really big peak telling us that it absorbed quite a bit, right? So again, this is at the same spot on the x-axis, lambda B, but we just absorbed way more light. So maybe over here we absorbed 20% of our light, and over here we absorbed 30% of our light, and over here we absorbed 40% of our light. We're going to use this information to make our calibration curve. So what these are here, these guys are spectra. When you have a spectrum, a singular spectrum, plural is spectra, you're going to have this graph of wavelength versus absorbance. We're going to take that and make a calibration curve. So I'm going to write cal curve. Your calibration curve is going to be a graph that you make in Excel, and I have a different video that'll show you guys how to make graphs. So you're going to make a different graph in Excel, and what you're going to put on this one is absorbance versus concentration. Okay, so we can see over here, this is our 1 molar concentration. We also have 2 molar and 3 molar. So 2 molar, 3 molar. So 1, 2, 3. And we saw that this was 20%, 30%, 40% absorbance, right? So if I go through here, I say, okay, 1 molar, here's 1 on my x-axis for concentration. I absorbed 20%. So I'm going to put a dot on my graph there. And at 2 molar, I absorbed 30%. So I'm going to put a dot on my graph at 2 and at 0.3. And then at 3 molar, I absorbed 40%. So I'm going to come and put another dot over here. And then we can go ahead and plot a line through these dots. And you can do that in Excel. Excel will do it for you. And what you get out of that is an equation in the form y equals mx plus b. In our case, we are going to tell Excel to make b equal to 0 because at 0 concentration, if there's nothing happening, you should not be absorbing any light, right? If there's no dye molecules, nothing that absorbs light, there should be no absorbance happening. So we're going to tell our equation that we want b to be equal to 0. So you're actually just going to have an equation in the form y equals mx. So remember, y here on our y-axis is absorbance. We have absorbance. M is just your slope. So that's going to be some number. I'm going to leave it as m. And x is going to be your concentration. Okay, so once you have this equation, if you take your unknown solution, so you're going to do this all twice, right? So you have all of these solutions of blue dye, all of these spectra for blue dye. Then you take all of that blue dye information and put it into one graph, which is going to be your calibration curve. You're going to do all of this 2 times, once for your blue dye, once for your red dye, or whatever 2 colors you have. Remember the example that we started with, we had blue and red dyes in our drink. So once we have all of this information done, again, we get 2 calibration curves. So let me erase some of this so I have room to explain the next part to you. Okay, so you end this whole process with 2 calibration curves. And from both of those calibration curves, we get an equation, right? So we had absorbance is going to be equal to slope times concentration. And this is for blue, right? And then everything I just said, you're gonna do all of that again for your red dot. So we're gonna get another equation where we have absorbance is equal to the slope of that graph times the concentration. And that's for red. Okay. We also know, if you remember when I was explaining all of the spectra for the blue dye, remember when we drew them, maybe the peak got taller, but it was always at the same place on this axis. So this was a spectrum, so this was the wavelength axis. So that means we know we have a wavelength for blue, which I'm going to call lambda B, and a wavelength for red. That'll be important in just a minute. When you put your beverage, you're going to take a little bit of your beverage and put that into the spectrometer. Because your beverage has two colors in it, when you get that spectrum out, you will see two peaks for your beverage spectrum. Don't be scared, that's okay. One of these peaks will have the wavelength for the blue dye or whatever dye, and one of them will have the wavelength for the red dye. So if you remember, red always absorbs at the same wavelength. So when you did all of your solutions of red, those are going to come out as one wavelength. And blue always absorbs at the same wavelength for blue. So all of those will come out of blue. So that's how you know which peak is which. Those wavelengths will be identical. What you want to plug into your calibration curve, maybe for your blue peak, you notice absorbance is like 0.25. And then maybe for your, oh, I'm losing my markers. Maybe for your red peak, you notice that your absorbance is 0.6. I'm just making up numbers right now. You are going to take these absorbance values. You're going to take this 0.25 and plug it in for your blue absorbance in that equation that you got from your calibration curve. And you're going to take the 0.6 and plug it into your equation from your red concentration curve. What this allows you to do, now you have an absorbance value for both. You have a slope. The slopes will be different, but they'll just be a number. And you have concentration, which you don't know. What you have now for each of your dyes is one equation with one variable that you don't know for. So all you have to do now is solve for concentration. So if you remember, if you have an equation y equals mx, and you want to solve for x, m is in the way, you're multiplying by m right now. So you need to divide by m to cancel it out. And because you did that on one side, you also need to divide the other side. And so that leaves you with x equals y over m. So you'll apply that same algebra logic to these equations and solve for your concentration. So we've got a couple of weeks to do this lab, and I know that was a lot of information that I just threw at you guys. I'm going to be there to help you through it, but that is your quick rundown. And also you will have this video available as a resource for when you guys are doing your lab reports on this lab. Thanks so much for listening.